Hello everyone, I am Satyasidas, recently pursuing my Masters in Zoology in Centurion University. Today I am going to discuss about the structure of immunoglobulin. So first of all, what is immunoglobulin? Immunoglobulins are the glycoproteins produced from the white blood cells. So what is glycoprotein? Glycoproteins are the product of glucose and protein. Glucose and proteins combines to form glycoprotein. So immunoglobulins are one type of glycoprotein. Immunoglobulins are produced during the acute immune system. So what is acute immune system? That immune system develops after the birth, that is known as acute immune system, or which are acquired by our cells, that is known as acute immune system. Acute immunity or acute immune system responds against the pathogen or the foreign particles or the antigens by primary and secondary response. They produces two types of cells or two types of lymphocytes, that are B lymphocyte and T lymphocyte. The B lymphocyte produced special types of protein against the pathogen. That protein is known as antibody or immunoglobulin. The T lymphocyte never form the antibodies. It helps the synthesis of antibodies by helping the B lymphocyte. But the T lymphocyte never involved in the directly synthesis of antibodies. So let's comes to the structure of antibody. Structure of antibody, as I discussed, that antibodies are one type of glycoprotein. They bind to a specific antigen. Different types of glycoproteins bind to different types of antigens. Roughly, the structure of antibody is Y-shaped structure that contains four polypeptide chain. Polypeptide chain means number of amino acids joined by the peptide bond to form a polypeptide chain. That that has one N terminus and one has C terminus. Four polypeptide chain or the four peptide chain can be divided into two types. Two small identical chains that are known as light chain, two identical longer chain which are known as heavy chain. Hence, that antibody can be represented by H2 and L2. The light chain of the antibody formed by either kappa or lambda. The antibody never possesses both kappa and lambda small polypeptide sequences. Either they contain kappa or lambda. The probability of kappa and lambda, or the concentration of lambda and kappa, varies from organism to organism. In case of human, the kappa polypeptide sequence have 60% concentration and lambda polypeptide sequence have 40% concentration. The differentiation of antibodies, or different types of antibodies, are produced by the heavy chain. Different types of heavy chains are there. They classify the antibody into five types. This is the structure of the antibody, rough structure of the antibody. As you can see that there are three regions, of antibody. What are they? F heavy region, then another one is hinge region, one is F heavy region, another one is hinge region, then third one is FC region. That F heavy region is stands from fragment of antigen binding. Where the antigen binds to the antibody, that region is known as F heavy region or fragment of antigen binding region. Then comes to the hinge region. Hinge region is the joining of two different types of heavy chain. That region is known as hinge region. Hinge region provides the flexibility to the antibody. Then comes to the FC region or fragment of crystallization region. This is known as fragment of crystallization as that contains the stabilized amino acid. That's why these are known as fragment of crystallization region. They form a crystal-like structure. That's why they are known as fragment of crystallization. FAV region binds to the FAV region binds to the antigen, different types of antigen, and the FC region binds to the cell surface where the antibody expresses itself. Suppose one cell binds to the, suppose this is a cell, this is an antigen. So the antibody binds to the cell surface like this. This region is known as Fc region, where the antibody binds with the cell surface. That is known as Fc region. Where the antibody binds to the antigen, that is known as FAB region. The specific naming of that region is known as paratop. Paratop is the part of antibody where the antigen binds. Antigens epitope binds to the paratope of antibody. This is a simple sentence. The epitope of antigen binds to the paratope of antibody. Paratope is the part of antibody. Epitope region is the part of antigen. Then comes to the next slide, where we can discuss the different region like variable region, constant region, and hinge region. So let's start from the variable region. As I mentioned that this is a polypeptide chain, so they have N terminus as well as C terminus. The N terminus is in this side and the C terminus is in the side. This is because this is the amino terminus, this is the carboxy terminus. This is the amino terminus, this is the carboxy terminus. The variable region of the antibody is found towards the amino terminus. The variable region it is called as variable region as different type of antigen binds to different types of antibody. The structure of antibody almost similar except the variable region. They vary antibody to antibody. Different types of antibody, different types of variable region, different types of antigen, different types of variable region. The variable region is found towards the amino terminus of the antibody, which is composed of 100 to 110 amino acid. About 20% of the variable region has so hyper variability, so maximum variability, and this region is known as complementary determinant region. As shown in the diagram, this is the complementary determinant region where the antigen binds to the antibody. They varies different organism to organism as well as different type of antibody to antibody. Then comes to the hinge region. The hinge, as I told that, hinge region provides the flexibility to the antibody like this. This is if this is a antibody, and this is the hinge region. If you move that finger, then this shows the flexibility portion. This is known as hinge region. The hinge region are the region where proteolytic digestion occurs. Proteolytic digestion means where the protein degradation occurs. The enzymes involved in the protein degradation is known as proteosome. That proteosome degraded the protein, that is known as proteolytic digestion. This hinge region is rich in lysine, cysteine, aspartic acid, and proline amino acids. Some antibodies shows hinge region and some antibodies lack the hinge region. The antibody which show the hinge region are immunoglobulin D, immunoglobulin A, immunoglobulin G. And that antibody which don't show the hinge region is known as immunoglobulin M, immunoglobulin E. The hinge region is found in between the constant region of heavy chain 1 and constant region of heavy chain 2. So what is constant region? Let's come to that point. In this diagram, we can consider that this is the variable region of heavy chain, variable region of heavy chain, that's why it is mentioned as VH. This is variable region of light chain, that's why it is mentioned as VL. This is the constant region of light chain and this is the constant region of heavy chain 1. Heavy chain contains 3 or more constant region. Constant region means they never change their sequences. Their sequence always same throughout the generation. There are number of antibodies, 5 types of antibodies. Their sequences never changes. That's why that is known as constant region of heavy chain. This is the constant region of heavy chain 1. This is the constant region of heavy chain 2. This is the constant region of heavy chain 3. This is also known as domain, constant domain of heavy chain. They never change their sequences. That variable, this is the variable region because it varies on antibodies to antibody depending upon their antigen binding. This is the hinge region. This one is the hinge region where disulfide linkage are present. This is present in between constant region of heavy chain 1 and constant region of heavy chain 2 as shown in the diagram. Then comes to the next point, that is constant region of antibody. Constant region of antibody contains 3 or more constant domain region in heavy chain and one constant region in light chain. As shown in the diagram, the heavy chain contain in this diagram 3 constant region, but the light chain contain one constant region. In heavy chain, that may be varies from 3 to 4 in number. For heavy chain, that is that can be written as constant region of heavy chain 1, constant region of heavy chain 2, and constant region of heavy chain 3. For light chain, that can be mentioned as constant region of light chain 1. Other regions of immunoglobulin. As I already discussed that FAB, what is FAB region, what is hinge region, and what is FC region. Then comes to the next slide, that is proteolytic degradation. As I already told that proteolyte degradation means degradation of the protein or the protein denaturation. That is caused by the different types of proteasome enzymes like papain, pepsin, mercapt ethanol. Papain is produced from the papaya. Papaya produces papain proteolytic enzyme that breaks the immunoglobulin or antibody into 3 fragments. 2 is FAB and 1 is FC region. As shown in this diagram, that papain breaks this bond. Papain breaks this bond. That's why it produces 2 FAB region or fragment of antigen binding region, 2 fragment of antigen binding region, and 1 FC region or fragment of crystallization region. Then comes to the next proteolytic enzyme, that is pepsin. Pepsin breaks that antibody into a single pad, that is known as FAB prime 2. Why it is called as FAB prime 2? Because it breaks after the hinge region. That pepsin breaks after that hinge region. That's why the FC fragment which are produced are fragmented into small particles. That's why they cannot be detected easily. Only that FAB regions are detected on the screening test. That pepsin digests the antibody after the hinge region or after the double sulfide bond. That antibody antigen binding region of antibody can bind to the antigen at their antigen binding site, because this is antigen binding site and this is a antigen binding site. That's why they can bind antigen as well as act against the antigen. That is used, that type of materials are used in the biotechnology process. Then comes to the mercapt ethanol proteolytic enzymes. That proteolytic enzyme, that mercapt ethanol breaks the disulfide linkage. Where are the disulfide linkage? They can break that disulfide linkage. Here is the disulfide linkage. Here one is disulfide linkage. Here 2 disulfide linkage. So it breaks that disulfide linkage. That's why it separate that light chain, this light chain, this heavy chain, and this heavy chain. As shown in the diagram, there are 2 light chains separated and 2 heavy chains are also separated. These are heavy chains are separated and these light chains are also separated. So 4 chains are found. When we detect their weight, the heavy chains, each heavy chains weight about 50 kilodalton, whereas the light chain weight about 25 kilodalton approximately. Then comes to different types of antibody. as I told that there are 5 types of antibody depending upon their heavy chain. First comes to the immunoglobulin A or antibody A. There are 2 types of isotope. Isotope means their structural is they are same, little bit different in their function. That are known as isotope. In immunoglobulin A or antibody A, there are 2 types of isotope. That are isotope A1 or immunoglobulin A1, immunoglobulin A2. In immunoglobulin A1, the heavy chains are present as alpha 1, alpha for A. That's why this is immunoglobulin A or antibody A. Alpha 1 heavy chain is present in immunoglobulin A1. Alpha 2 heavy chain is present in IgA2 or immunoglobulin A2. In light chain, either that kappa or delta. Sorry, either that kappa or lambda light chains are present in immunoglobulin A or antibody A. That immunoglobulin A or antibody A is found in 2 form in human being. One in the form of serum and another in the form of secretory form. In the secretory form, when we consider the secretory form, immunoglobulin A is secreted from the human body. When a mother feeds her baby, that colostrum contains the immunoglobulin A. That form is known as secretory form. When we consider the blood serum, that in blood serum, the immunoglobulin A is found in the serum form. The molecular weight of the serum immunoglobulin is 160, and the secretory immunoglobulin A has the molecular weight of 385. Why this is heavier than the serum immunoglobulin A? Because this immunoglobulin contain the secretory component as well as the joining chain. Because they are found in the dimeric form. Dimeric form matlab two single structure joined through a joining chain. That complete structure is known as dimeric structure. Monomers, two monomers combines to form a single dimeric structure. The serum immunoglobulin A is found in the monomeric structure and the secretory immunoglobulin A is found in the dimeric structure. They are found or appears in the mucosal area such as the gut, respiratory, urinogenital tract and prevent their colonization of the pathogen. They prevent the colonization, forming of colony like bacteria. They prevent their colonization in our body. Then comes to the next types of immunoglobulin, that is immunoglobulin D or antibody D. D for delta. The heavy chain have delta polypeptide heavy chain. That's why this is named as immunoglobulin D or antibody D. The light chain either kappa or lambda. The molecular weight of the IgD or immunoglobulin D is 180 kilodalton. It is found in the monomeric structure in the human body. That function is unclear. That which type of function it occurs, that is unclear till now. It act with the IgM, immunoglobulin M type. That is another types of antibody. With IgM, IgD act with B cell. B cell development, in the B cell development, IgD works with IgM. Then comes to the next type of immunoglobulin or antibody, that is IgE. E for epsilon. The heavy chain of the IgE antibody is epsilon. The light chain either kappa or lambda. The molecular weight of the IgE immunoglobulin or antibody is 188 kilodalton. It is found in the blood serum in the monomeric structure that binds to the allergens and triggers the histamine release from the mast cell. So IgE antibody is known as allergic antibody, because when some allergic condition, itching, sneezing, these type of symptoms are found in human body, that time suddenly IgE or at first IgE antibody is synthesized. They synthesize the histamine. Histamine causes the allergic response from the mast cell. That's why this is also known as allergic immunoglobulin or allergic antibody. Also, they protect against the parasitic worm. Different types of parasitic worms are there. It prevents their growth in our body. Then comes to the immunoglobulin G. There are four types of heavy chain in case of immunoglobulin G or antibody G. Depending upon the four types of heavy chain, there are five types of isotope of immunoglobulin G or antibody G. In case of immunoglobulin G1, IgG1 heavy chain is gamma 1 and the light chain is kappa or lambda. The molecular weight is about 146 kilodalton. This KTA indicates that kilodalton. This structure is found in case of in the form of monomer, and this is divalent. Divalent matlab monovalent and divalent. This can be act with two different types of antigen at a time. It can bind two different types of antigen at a time. That's why this is known as divalent. All the isotopes of immunoglobulin G are monomeric and have divalent in nature. All the antibody G or immunoglobulin G can pass the placenta from it can transfer from mother to offspring through placenta except immunoglobulin G2, that is G2A and G2B. They cannot pass the placenta. That's why other rest of the three isotopes of immunoglobulin G can pass through the placenta and protect the body and protect the baby from outer injury or different types of disease, and increases the immunoglobulin G or antibody number in the human baby when they are in the uterus or they are in the womb. Then comes to the isotope or the antibody immunoglobulin GM or IgM. They have a heavy chain mu for m, and they have light chain either kappa or lambda. The molecular weight of the IgM is 970 kilodalton, because it is found in body in pentameric structure. Pentamer structure I'll discuss in later, or shown the next in the next slide. I will show the diagram. That is pentameric structure. IgM is found in the blood in pentameric structure. The fast response of the antibody is the IgM. When any antigen enters in our body, the fast antibody interact with that antigen is IgM, that is expressed on the surface of the B cell and is secreted with the very high avidity. The ability matlab high concentration, in high concentration IgM secreted from the B cell. As we already discussed that all the type of immunoglobulin, all the type of antibody are synthesized from the B cell. When the B cell matures, when the B cell act against the pathogen, they divided into two types of cells, that are plasma cell. Another one is memory cell. Plasma cell interact directly with the pathogen when the antigen or pathogen enters into our body. The memory cell remember the specificity of that antigen or that pathogen and act when that antigen enters in our body second time. The memory cell act in the second time, or that memorize the specificity of that antigen. This is all about the different types of antibody. Then comes to the structure. This is the structure of IgG. This is monomeric structure. This is IgE or immunoglobulin E. This is also monomeric structure, but they, as I told that in IgM and IgE, there are no hinge region. There are no hinge region. Single sulfide bond is present. Here disulfide bond is present. Here hinge is present, but there is no hinge present in between IgM or IgE. This is monomeric structure. This is also monomeric structure. This is also monomeric structure. This is dimeric structure. This is pentameric structure. 5 unit monomeric unit join through the joining chain, and that form IgM structure. This is IgA. This is dimeric structure. Two monomeric unit join through the joining chain, and they can secrete from the mother's mammary gland. That's why this is also known as secretory protein. This is secretory immunoglobulin, and that protein is known as secretory protein. This is all about the structure. So thank you. This is all about that